In lower organisms, including yeast and Drosophila, the Golgi apparatus exists as discrete stacks of cisternal membranes scattered throughout the cytoplasm. In vertebrate cells, however, individual Golgi stacks fuse into a continuous ribbon that clusters around the centrosome. Why the vertebrate Golgi is organised like this is a question that fascinates Rosa Rios from the Andalusian Centre for Molecular Biology and Regenerative Medicine in Seville, Spain.
We know that the pericentrosomal Golgi ribbon organization is not critical for protein modification or for global secretion. So the question is why vertebrate cells prefer a Golgi ribbon to individual stack. Another question is why is this ribbon located around the centrosomes? These are the questions we wanted to investigate with this study.
To address these questions, Rios and colleagues, including Lydia Hurtado, Cristina Caballero and Maria Gavilan, investigated a protein called ACAP450. This large coil-coil protein localizes to both the centrosome and the Golgi, and it maintains an intact pericentrosomal Golgi ribbon by helping to nucleate microtubules on Golgi membranes.
The sequence responsible for targeting to the Golgi was unknown. So we wanted to identify this sequence in order to interfere with the Golgi-associated pool of the protein.
Hurtado et al. Made a series of truncated versions of ACAP450 and found that a large N-terminal fragment, AK1, localized to the Golgi by binding to the Golgi resident protein GM130. The researchers then truncated AK1 even further and identified a smaller fragment, AK1b, that could also bind GM130 and target to the Golgi.
And when we express this fragment, the endogenous protein is displayed from the Golgi. And in both cases, the microtubule nucleation on the Golgi was inhibited.
Surprisingly, however, the two fragments had different effects on Golgi positioning and morphology. The smaller construct, AK1b, broke the Golgi ribbon into fragments that remained clustered around the centrosome, a phenotype also seen when ACAP450 is depleted by RNAi. In the presence of the larger AK1 fragment, on the other hand, the Golgi ribbon remained intact, but it was no longer tightly associated with the centrosome.
We didn't expect this different behavior of these two mutants.
After mitosis, or after washout of the microtubule depolymerizing drug nicotazole, the Golgi ribbon reassembles via a two-step process. In the first stage, Golgi elements fuse together in the cell periphery, a process dependent on Golgi nucleated microtubules and which is therefore called G stage. In the second step, the Golgi membranes move to the cell center along centrosome nucleated microtubules, a process called C stage. In cells expressing AK1, peripheral Golgi fusion occurred, albeit very slowly, but the Golgi ribbon failed to translocate to the cell center and associate with the centrosome.
So this result suggested that although less efficiently than in control cell, the G state can take place, whereas the C state is inhibited. It's for this reason that the Golgi ribbon formed in random location.
AK1b, on the other hand, caused Golgi fragments to quickly cluster in the cell center, but peripheral fusion was inhibited, suggesting that AK1b blocks G stage but allows C stage to proceed.
Our data indicate that if you selectively perturb one of the two stages of Golgi ribbon formation, the effect on Golgi organization are completely different.
How AK1 and AK1b selectively perturb the different stages of Golgi assembly is unclear, but Hurtado et al. Found that one difference between the ACAP450 fragments is that AK1 can bind to the dynein-dynactin subunit P150 glued, and can therefore interact with microtubules.
So the large AK1 fragment is a dimer containing Golgi and microtubule binding site, whereas the short AK1b dimer only contains the Golgi binding domain.
Whatever the mechanism, AK1 and AK1b's different effects on the Golgi's organization allowed Rios and colleagues to investigate the importance of pericentrosomal positioning and ribbon integrity to the Golgi's functions.
So we evaluate the contribution of positioning versus integrity in cell polarization, in secretion, in migration, or ciliogenesis.
Global secretion was unaffected by either fragment, but both AK1 and AK1b inhibited the polarized secretion of neuropeptide Y at the leading edge of migrating cells, probably because both fragments disrupt Golgi nucleated microtubules, which are required for directional vesicle trafficking. Hurtado et al. Then looked at cell polarization in response to wounding.
At different times after wounding, we compare the reorientation of the centrosome and the Golgi toward the leading edge in control or in transfected cell.
In control cells, the Golgi and centrosome reoriented to face the wound edge within 5 hours. In AK1b expressing cells, in which the Golgi is fragmented but still associated with the centrosome, the organelles took slightly longer to polarize. In AK1 expressing cells, however, where the Golgi ribbon is intact but dissociated from the centrosome, reorientation of both organelles was inhibited, suggesting that Golgi pericentrosomal positioning is required for cell polarization.
We performed the same wound healing assay and we tracked individual cells and we detected that cells expressing AK1 were able to move, but they moved in the wrong direction. On the contrary, the AK1b fragment showed reduced mobility, but without any obvious effect on the directionality of migration. So it was the cells expressing the large AK1 fragment were more effective in migration, in my opinion, because the centrosome and the Golgi are not well oriented. The cells are not well polarized.
Finally, Hurtado et al. Looked at the ACAP450 fragment's effects on ciliogenesis. Both AK1 and AK1b strongly inhibited the formation of primary cilia, suggesting that both pericentrosomal localization and Golgi ribbon integrity are required for cilia assembly.
I think that we have come to two major conclusions. So first is that pericentrosomal localization is a more determinant factor for establishing the cell polarity axis and directional migration. And the second is that the formation of a primary cilium is critically dependent on both proper Golgi location and integrity.
These essential functions in polarity, migration and ciliogenesis may be why the vertebrate Golgi evolved into a continuous ribbon surrounding the centrosome. Rios and colleagues now want to investigate how the association between the Golgi and centrosome is regulated during mitosis, when the two organelles are separated and the Golgi fragments. ACAP450 may be a key target for mitotic kinases during this process. But in the meantime, you can read more about the different contributions of Golgi localization and integrity in the paper by Hurtado et al., published in the May 30th, 2011 issue of the Journal of Cell Biology.